or the spore print?
The spores. Yeah, so
Okay.
I've got my spore print. I'm just gonna like press it down lightly onto the surface of the auger and seal it up.
Okay.
Just kind of a little tap is all you need. Your goal is to get 20 or 30 spores. You'll have thousands of them just by doing a little tap. So,
okay,
it'll work.
Do a little tappy tap.
Yeah, I found these super cool red mushrooms with these like white bumps on top that I've been growing. So
I know that's a lie because those are obligate biotrophs. Fortunately, there are cultures around the world, people who do eat those, the fly agaric, the red with white spots. And apparently the trick is that you dry them out thoroughly, like 100% dry, and that kind of deactivates some of the toxins, or most of them, and then you eat the mushrooms, and then you collect your urine afterwards, and then people drink your urine and hallucinate. Yeah.
Okay, that's what I was gonna ask. I was like, it's gotta be, if anybody's drinking piss, it's gotta be to get you high.
Yeah. I've never tried this, but from like ethnographic accounts and everything, it doesn't seem like it's getting high or like it's fun so much as like a religious sacrament for some groups. You'll find it native peoples like the Sami people in northern Sweden and stuff have a long practice of eating mushrooms like that, or drinking mushroom piss like that, I guess. Kind of weird, but to each their own. Still, to me, it seems pretty dangerous. Anything from the Amanita genus should really be off limits as far as human safety. So do not constitute this as an endorsement of eating those mushrooms and then drinking pee. All right.
I had a question about one of my spore prints.
Yeah.
It did not go as well as my first spore print, and I think what actually happened was just the gills just ripped off and stuck to the tinfoil.
Oh, yeah.
Because they were like really wet when I put them down.
Mm-hmm.
And I think that the gills just came off on it. Do I need to like just trash that and find a new mushroom?
Can you see the color of the spores?
Yes.
That's the important thing from the spore print.
Okay.
So you'll be okay. The reason we do spore prints when identifying mushrooms is it gives us an accurate look at the color of the spores.
Is the color not just the same as the gills?
Not always. No.
Oh, okay.
Yeah. So a mass of spores will have a different color than the gills sometimes. Then you take some of those spores, throw them on a microscope slide, and you can see their shape and size, and that's a really good character for identifying macrofungi. And you'll note that in the guidebooks, even like the Audubon Field Guide to North American Mushrooms. You find the mushroom, it looks the same, then you flip to the back of the book, and there's a section on spores, and it'll tell you the spore print color, and then describe the spores, how big they are, what shape, whether they have any ornamentation on them. And for a lot of basidiomycetes, you're going to have to get down to that if you're looking for species level identification. You pretty much have to go to the spores under a microscope.
Okay. Also, for the pollen.
Yeah.
What am I supposed to be seeing in here? Like, or are we just taking the water after it's been sitting for a while and putting it on? Okay.
Yeah, it's microscopic, so you're probably not going to be seeing anything until you stick it under a microscope.
Because I thought I saw something on the instructions like you should see like a film or something.
It looks like a little, like a pudding skin almost, kind of. It just gets kind of filmy if you got strong growth.
What
I
do you
apparently do not have strong growth.
Is it yellow?
The pollen is yellow. The rhizoids from the chytrids is clear, and so it just kind of looks like a thin, filmy skin. Man, it's hard to describe.
Because it looks like I have growth on the top, but I can't, like, it's kind of yellowy, kind of clear as well, but I can't tell if that's just like the pollen grains or if that's actual growth.
The yellow you're seeing is the pollen grains, but if
Okay.
it looks kind of like congealed or like there's a kind of a film almost, probably the pollen grains are being eaten and you're good to start pulling some out and looking under a scope.
Okay.
Yeah.
I think,
if we
I think
can't
somebody
see-
posted a
oh.
picture of one too on the Teams page. You can
Yeah,
see.
we do have a good picture of kind of what you're looking for on the Teams page in our general, I believe. If I scroll up. Yeah, Chandler sent in a nice photo, and I've kind of like annotated it with some arrows showing you what you're looking at.
And to be, excuse me, and to be absolutely honest, I just took like several pictures of different parts on my slide because I wasn't like 110% positive at what I was looking at, and just sent it to Doctors On because I didn't know exactly. It's just like really concentrated on mine. So
Yeah.
I also put some extra water drops on it to try to spread them out a little bit, but that helped.
So if we can't find anything, can we go back and put more water in from like a different source and it will still work, or how does that all work?
If you haven't used up all the pollen in there. So I've got a little tube of pollen in my lab, and I just stuck a Q-tip in it basically, and then tapped it over each one of those tubes. So there's some pollen. I didn't like count the pollen grains or anything. You may have used it all up or like dumped it on your slide. It's unlikely because the plastic actually kind of has some hydrostatic force, and the pollen wants to stick to the edge of the plastic when it's in there. So it's worth another shot of finding some more water and throwing it in.
Okay.
Barring that, you can go get yourself some pollen
Where do you get that?
from plants.
Okay.
I don't know. Angiosperm pollen doesn't work as well, but it's pretty good. So you could go like anything that's flowering right now and just either rip off a couple stamens and throw it in, or just tap it over your container. Pine pollen is not out and about right now. That's a springtime thing, so you're out of luck for pine pollen.
So I did it a couple weeks ago and I didn't see anything. Is it still going to be good even though I put it in a couple weeks ago, or is it kind of?
Yeah,
Yeah.
I think it's the even when we do this in the classroom, like in a lab setting, everyone's stuff grows at different rates because they all have different water sources. It's almost impossible to predict how long it'll take for things to colonize, or if you did actually have chytrids in there, that kind of stuff. So
Okay.
I also have like a little growth above the water. Is that okay? Like it looks like the tube is okay. It's a little like opaque, I guess.
yeah.
Okay.
I mean, scrape some of that off onto a slide. And take
All right,
some
and that should be
some
some of
pictures
it.
of it. Yeah.
Okay, great.
All right. Shall we chat about this paper now?
All right, last thing, Doctor's On,
Okay.
before
No
we get in the
worries.
paper. So I did the whole mushroom or the macro fungi assignment, but now that we're talking about the spore prints, I remember I sent you an email that had like pictures how mine didn't quite show up. So I kind of used like instead of using a marker, I used like an ink pad to be able to show the print of it, you know.
Mm-hmm.
But I don't think I can really get any spores off that now because I just
Because they're all
bought them on the mushrooms, you know.
ink blotted.
Because you said you could use like a Sharpie, you know, to kind of help it show up. So I was like, well, I'll just use this. It's even better, you know.
Yeah.
I'm a beginner at
The
this.
Sharpie, yeah, I guess I should have been more clear. The Sharpie is for the foil. So you darken a patch of the foil
Oh, well.
Yeah.
I
So
thought you were saying to darken the bottom of the mushroom. I was like, all right, I'll just use.
no, I like to do half like white, half dark, and so I
Yeah.
Sharpie half of it and then set the mushroom down over the whole thing. And that way, no matter what color the spores are, they're going to show up against the contrasting background.
Well,
So,
I have great pictures of the. It came out great, but I don't know if I've got too much color besides black. I have one that turned out great by itself, but the other 4 are gonna mostly be black.
and that black is from Sharpie, I'm guessing.
Well, the black is like literally from the ink pad because I did them normally, like I sent you the picture, but they all just came out white and I couldn't see anything. And so I figured, well, I got to go to black.
That's awesome. I did not anticipate that. Okay, yeah.
Well, when you said the Sharpie, that's what I was like, I'll just take it at the next level and make it even more clear, is what I was thinking. But
all right, what you've done is a really cool art project, but not necessarily gotten spores. You've gotten a print of the gill structure, which isn't without worth, and probably looks pretty cool. But what we don't have is the actual color of the spores, and that's kind of the character we're looking at. It's the spore print, because the guidebooks are all going to be like spore print white or spore print light cinnamon color, that kind of thing. So
even if they were all white beforehand on the papers, like if
well,
I included all, like, would that
if
not
they're
count?
white, then you've got that information already.
So then I would be more or less good to move forward?
Yes.
Okay. Okay.
Yeah.
Because I did it on the papers and they were all white, and so I was like, I'll do it the other way, and then of course I don't have it, just ink now. So.
Right.
But okay, thanks, Arzon.
Okay, yeah, just don't accidentally think, if you're trying to key this out or something, that the spores are black.
Right. Yeah.
Right.
Definitely not black. Thanks.
Okay.
So the purpose of doing the circle with half of it colored in with the Sharpie is in case those spores are white. Is that why?
It shows up way better against a black background.
Okay, that makes sense.
Mm-hmm. That is hilarious. I'll make sure for future times I teach this to be real clear about that. Okay. Oh yeah, the paper. I think we're ready. So let's talk about it. Who wants to start?
Give me a second.
I have a question. I have a question about, so it talked about like yeast coming from like filamentous ancestors. So what would be the purpose of them kind of, it seem like they're going backwards to just single cell. So why would yeast do that?
I'm gonna turn this around and ask you, can you think of any reason that would be selected for evolutionarily?
Yeah, because the point of the paper was kind of talking about how they were multicellular, but then re-evolved to be unicellular, and why. And I don't know, other than just saying that was the ideal way for it to capitalize on its environmental niche, but.
Yeah, I mean, sorry.
I was gonna say, it seemed that the paper was suggesting that yeast can turn on the genes, basically. That's why I'm assuming that's probably when they're under stress, whether it's just reaching nutrients or whatever. And because it makes sense that if you can't really move and you're a unicellular organism, but you can then start growing hyphae, you can reach farther. You can grab more stuff. So that's, I guess, my understanding.
Yeah, so the paper did point out that all these yeasts do seem to retain a lot of the, maybe not all of, but at least a lot of the multicellular associated genes. And we have seen yeast form what we call pseudohyphae, where yeast are just like budding but staying connected, essentially. So they're still kind of doing it. as to why they would have lost hyphae to begin with, can anyone think of a plausible explanation?
So this is way out there. This is coming up.
Great.
With. First, let me ask, what evolved first? Was it septa or yeast?
Let me think for a second. Septa.
Okay, so let's say there is a fungus with some septa, all right? Some septa broke off, and now there's only one nucleus in there. That thing just kept growing, and eventually it was just like, well, I need to reproduce. So it kind of just made another one of itself, which would have been budding since it was single cell, and continued on from there. And now you have yeast. Something along the lines of that.
Okay, I can see that happening. Would that be immediately selected against by natural selection, or if not, in your scenario, obviously not, what would be advantageous about it? That natural selection would see that and say, oh, good job.
Well, if it was small enough, you'd have a nice surface area to size ratio, but that's what filaments are for anyway, the hyphae.
Mm-hmm.
So maybe it's not that it necessarily was selected for, but that it wasn't selected against.
That's where I'm leaning. This is a gut thing. I don't know that we have evidence either way on this, to my knowledge. But my gut would be that maybe it wasn't selected for, but it wasn't selected against. Because a yeast single cell still has a good surface to volume ratio, just like hyphae do. They can dispense with some of the energy costs associated with like long distance transport of things, which the paper kind of talked about, touched on at least. So maybe it's evolutionarily just okay to be a yeast. But the paper did point out that since a lot of the multicellular genes are still conserved in yeast, they haven't been reduced beyond functionality, that there probably is still some evolutionary reason why that's the case. So something advantageous about being able to form pseudo-hyphae every now and then. And I think Christina was saying just a minute or two ago, like, it kind of makes sense that hyphae are useful because you can get more nutrients, you can like get into spaces, surround things, use it to grow into substrate, that kind of stuff.
Dr. Zahn, so are we saying that those yeast have genes like, in their introns or something that would, technically maybe allow them to grow hyphae if stresses, were applied and mutation happened or something, that they could turn back into a hyphae producing organism?
Yeah, that's exactly what we're saying. And we do see it. We even see it with like brewer's yeast, Saccharomyces. In certain conditions, they will form pseudo-hyphae. We're not exactly clear on what triggers that, or if it is advantageous for sure, but we do notice them doing it from time to time in culture. They moved from kind of a single-celled to a colonial multicellular form, expressing those genes that are dormant.
Do they ever just like fully like turn the corner though and just stay that way?
In baker's yeast, no, but in important human pathogens, we'll see at least one today, they do. And so Candida is kind of the classic example of this. Candida lives in our mouth, on our skin, all over our bodies. and it is in a budding yeast form most of the time. If you are immunocompromised, though, it will start growing hyphae and devouring your body. And so the hyphae are kind of a mark of it's turned pathogenic. Part of my postdoc work was in plant pathogens for endangered plants, and I saw the same thing. There was a yeast, pseudozyma, and it lived on the surface of plant leaves as a yeast, basically doing nothing. But when spores of this powdery mildew landed there, it detected the spores through some sort of receptor, I have no idea, but it could tell that they were there, and it would just suddenly turn into a hyphal growth form and attack the spores and eat them. So it can and does happen that a yeast will go totally hyphal.
And the paper said that they preferentially kept those genes. Do we know why that they say, or what mechanism keeps them? Because it's obviously not random.
Yeah, it's not random. We know that, and the mechanism presumably would be natural selection. But as for what natural selection is selecting for environmentally, no idea. This paper is really recent, and they did a good job in their background research and couldn't come up with a reason other than to say it's not random, and so it's probably being selected for by natural selection. It's probably adaptive, but we don't know what would be adaptive about that.
Dr. Zahn, I had a question though. Do you know, did multicellularity develop around the same time in fungi and eukaryotes, or was it eukaryotes and then later fungi, or?
Fungi, they are eukaryotes.
Oh, sorry, I'm sorry. When I meant I meant to say animal cells or plant cells or.
Oh.
Histocons, as you call them.
I think fungi actually got there first.
I mean, clearly it talks about the two different mechanisms, but.
Right. So yeah, fungi, we had like zygomycete stuff. Well, multicellularity in animals goes way back to, choanoflagellates and colonial stuff. So yeah, roughly the same kind of time frame.
Okay.
Yeah.
I mean, I know it's hard to find fossil record of fungi.
Exactly.
yeah, I get it.
Especially of microscopic fungi.
Okay,
yeah.
But assumed to be about the same time. Okay.
Mm-hmm.
That was one question I kept having. I know I have another question, but I'm trying to find the part.
Okay.
And, I'll be back in a bit.
I had a question. I guess it's more like vocabulary based. In the introduction, it talked about clonal versus aggregative mechanisms.
Okay.
What do they mean by that?
Well, this is something we don't cover. Diastelium. It used to be studied by mycologists, I guess kind of still is. Slime molds. Are any of you familiar with slime molds? Well, they're protists.
Just what you showed us in 16.
Yeah, only as far as
Okay.
about.
Yeah, so they're amoebozoans, that's their eukaryote supergroup, and they exhibit multicellularity as shown here. View image. And it's aggregative. So it's a bunch of single cells, you can see them on here, coming together and just like stacking on top of each other, essentially. And that's aggregative multicellularity. These things then move around like a little slug. It's multicellular. It looks like it has behavior and everything, but it's really just a bunch of individual actions by the cells that make up the body. They're not stuck together in the same way as clonal multicellularity, something like the water nets. Water net. Hydrodictyon, that's it. We've got clonal multicellularity. Okay, so they're budding off, staying there, forming a body out of that shape. Same sort of thing for corals, right? It's a clonal multicellularity. So the difference is, are you building a body that started small and grew out of itself, or are you building a body, multicellular body, by a bunch of tiny cells coming together and just stacking on top of each other, which would be the aggregative form. So which one are fungi, according to this paper?
Aggregate? Or no?
Neither.
Neither? Oh, okay.
Yeah, they're a new way of being multicellular.
Okay, thank you.
Yeah. Did that clear it up? I don't know if I was explaining that very well.
No, yeah, it was essentially just needed like the vocab to it.
Okay. Yeah, with aggregative, a lot of protists do this. It's just a bunch of cells kind of stacking on top of each other, and we see it and we think, ah, it's multicellular, and it can actually have multicellular features, and it can act like a single organism and big body and everything, but it's just cells stuck together.
I have another terminology question.
Okay.
It's at the very end of the abstract. It says co-option and modification of ancient eukaryotic functions. I thought those two things were the same. I thought that's like what co-option was finding a new function for old genes.
Yeah,
basically.
I think the term modification, you're right. The term modification in this context, though, would be that they co-opted, they found a new function for it, but they've also, that gene has undergone selection subsequently. And so it looks different
Oh, okay.
than it did before. So they're using an old gene to do a new thing. And since it's been doing this new thing for a while, it's actually accumulated mutations and been modified.
Okay, gotcha. Thank you.
Yeah.
I have another terminology question.
Okay.
It's in the paragraph about the phagocytotic genes being
Okay.
adapted. And it talks about the Wiskott-Aldrich syndrome family of proteins, the like WASP family being conserved.
Oh, yeah.
A family being, I don't know what those are. Can you kind of just
I don't know what those are either.
Oh, okay.
Yeah.
I was looking at figure 4 and I was like, I don't understand what this is trying to say.
Okay, so in the paper it says they reorganize the actin skeleton during phagocytosis. All right, so phagocytosis, right? You get your cell, it divots in, things get brought in a vacuole, so it's eating stuff essentially. It's kind of the cell engulfing and swallowing something. And so those WASP proteins apparently are moving around the actin cytoskeleton, making it possible to change the conformation of the cell. I don't know how they work, what they do, like what kind of domains those proteins have, but they're apparently a thing. If you're ever in a quandary and you need to know more about a certain class of proteins, KEGG pathway, you can go to KEGG pathway database. This is the Kyoto Encyclopedia of Genes and Genomes. So here, let's just see if WASP works. I'm going to actually try Wiscott Aldrich instead, because I'm not sure what that's giving me. Nothing. Oh. Interesting. All right, there we go. Okay, so here are the pathways it's associated with. Here are the diseases it's known to associate with. Here is where it's been found in things and the known genes in this gene family. You can click Show All to see a bunch of them. It gives you a reference. So we can click on taxonomy and kind of see where these genes fall evolutionarily. Okay, so they're found in Homo sapiens, Platarini. All right, so you can investigate any unknown gene in this way. And if you want to look at a specific one, since this is a family of genes, so here is that particular gene, things it's associated with. Okay, so associated with Yersinia, that's the Black Plague. All right, so interesting. Here's the nucleotide sequence for it, so you can throw this into a gene predicting program. It'll kind of show you the shape, the predicted shape, that kind of stuff. So just a little side note, the KEGG website is a super nice resource for learning about new proteins. All right.
Okay, I think I remember what my question was. So they talked about the paper about duplication of genes. It was a little bit confusing to me because they talk about how it's important, but then they go back to, but this is not very important. And I think also because they refer to different aspects. So I mean, it makes sense. But I guess my question was, has duplication of genes, is that one of the major hallmarks of multicellularity in animals? Or, I mean, why do they refer back to that all the time? Okay, so that is,
Yeah.
Okay, so that was something that they were looking specifically at to see if it does compare with other animal cells, I guess.
Absolutely, it's a major hallmark of animal multicellularity. Think about Hox genes, right?
Okay.
For body
Yes.
plan organization.
Okay.
All those are just duplicated. So all these duplicated genes lead to paralogs, right? So you can duplicate genes. Once you've duplicated it, you're free to kind of experiment through evolutionary time with one copy of the gene, okay, without losing the function, the original function. And so it kind of gives you like a little safety net. So you can still live and do the things your parents were doing, but you're free to kind of have slight differences in the other one, and it's free to wander around through evolutionary space, maybe hitting on something unique and interesting.
That makes a lot more sense what they're saying. So we see some duplicates, basically, a doubling of the genes, but it was not the main development, basically. That basically allowed the fungi to then evolve, I guess, through a mutation or whatever that led it to where we are today.
Yeah, their argument here is that the evidence seems to show that duplication wasn't as important as co-option and modification.
And exaptation, yeah. I saw that,
Yeah.
Okay, perfect. Thank you for that. That really helps. Yeah, because I kept wondering, I was like, why are we referring back to this? But then they're like, but it's not that important. I don't know.
Yeah, gotcha.
Thank you so much.
In particular, the phagocytotic genes exapted for hypomorphogenesis. This is pretty interesting. I mean, the claim here, and pretty good evidence backing it up, is that in things like amoeba and really early proto animals, right, before they were quite multicellular metazoans, cells were doing phagocytosis, and fungi can't do phagocytosis. They do not have that ability, but they still have phagocytosis genes. They're just using them to, instead of indent and surround a food particle and bring it in, they're using those same genes to grow forward, basically turning them upside down into hyphal production genes. So that's kind of fascinating to me. Is that hyphal growth, in some sense, is like the opposite of phagocytosis, and it's not. It's much more complicated than that. It's the same sort of genes just being used for a slightly different purpose.
Yeah, I was going to ask about that. Are they using those phagocytosis genes as part of the where they digest themselves at the end to grow?
No, that's a whole different thing. So they're using digestive enzymes for that. Yeah. So like chitinases, that kind of stuff, to weaken the chitin structure, or at least prevent it from being cross-linked. I see a hand from Braden.
Yeah, so it's kind of a related question to the paper and whatnot, but I remember in 1620 you talked about slime molds and whatnot, because it's kind of just an interesting thing. Are those classified as an actual fungi?
Mm-mm. They're mostly in amoebozoa.
Okay, so would they then follow kind of like the normal, typical, just a collection of multiple cells that kind of makes multicellularity? So
Yeah,
kind of like
they're aggregative in their multicellularity.
Okay. So they follow that path versus the fungal.
It mentioned that in the paper. You wouldn't know unless you knew this organism, but in that same paragraph about phagocytotic genes, we examined the phagocytotic genes in filamentous fungi based on the phagocytotic machinery of D. discoideum. That's the classic slime mold lab rat right there. So they're looking at that one in particular because amoebozoa is sister to fungi and the animals. So they're pretty close in the big picture evolutionary tree of eukaryotes.
All right, cool, thank you.
Mm-hmm. What about their methods? Was anyone able to follow those?
Kind of. Let me review.
So they built a phylogeny. Okay. Then they did ancestral character state reconstructions. So looked at on that phylogeny, is it hyphal or not, essentially. Okay. Using a Bayesian Monte Carlo Markov chain reconstruction. Okay. And then looked at gene family evolution. So they did that for, what was it, like 400 and something, 432 genes, or I don't remember. So they basically built phylogenies of each of those genes, mapped it onto the phylogeny that they built of the organisms. Then they found and tested genes associated with multicellularity, and then did a subset for just the phagocytosis. And then they did genome-wide screens for hyphal-related gene families in all those organisms.
Do you know what an all versus all blast is?
Yes.
Okay, what's going on there?
Let me show you. So BLAST is an acronym for basic local alignment search tool, and it's a way to look for similarities between nucleotide or protein alignments. Okay. The underlying algorithm is actually somewhat complex, and it has like penalties associated for gaps or mismatches or certain number of matches in a row, kind of boosts the rating. And so a blast is shorthand for saying we took the gene sequence we found and searched it against all known and reported gene sequences that are in this giant US National Library of Medicine repository. Okay. So in all versus all, that means we took every gene that we were interested in and blasted it against every other possible combination of those genes and against everything that we know online. So for this, let's see. We can go back down here to this nucleotide sequence, copy it. This is one of those WASP things, and just paste it in here in the blast and say, okay, what do we want to blast this against? Let's do the whole nucleotide collection, right? We could say like just the sequence read archive or just human genome associated stuff or whatever, but this one means everything. And so then we're going to just click blast, looking for highly similar sequences. This takes a few seconds. If you're doing this for 400 and something genes against everything, you're going to automate this with some computer code. But there we go. Here are the genes that match, at least quite closely, this sequence for this particular gene, which is, well, it's got a bunch of different gene names, but here they are. And yep, we're finding them. So BLAST worked for us. We took random, well, not random, but a crazy collection of A, T, C, and G in order, did a blast. Government supercomputers run this behind the scenes, and then they return all the hits that you found. So you can look for genes that are similar. These are 100% identity here. So those are all the exact same gene. Here is one that's 99.54% identical, and what's that? Pan-peniscus wasp actin nucleation promoting factor.
Oh, that's the one in the paper, right? WASP?
Well, that's, yeah, all these genes are, it correctly told us that this sequence was WASP genes. Okay, and it's showing the alignment. Here are the differences between the one we searched and the subject that it found a match on. So there's a difference right there. There you have it.
Is more genetic information added to the slide?
Nope. A G has changed to an A in this version, and that was found in this organism, Pan-peniscus.
What I meant was, is BLAST, like this database, is it updated? That's what I was trying to.
Oh yeah. Mm-hmm.
Okay, so it is constantly being updated. That's awesome.
Constantly, yeah.
Really awesome. Wow.
Literally millions of entries every day.
Perfect.
Yeah. And so sure enough, the one that we found in our keg here came from a human, right? And we've got an identical, except for one base difference in bonobo. All right. So in that way, we can actually look at the phylogeny of single genes and kind of like see how they're related in different species. And that's what they were doing in this paper, just with fungi and protists. Okay, so they were taking these genes, blasting them against all known gene databases ever, finding things that were really close matches and then essentially counting up the differences and seeing where they were in that phylogeny. So they built their phylogeny. Where are you? All right. So they build their phylogeny based on whole genomes to start with. And then for each one of those 430 different proteins, they mapped them onto this phylogeny, essentially. And so they said, okay, well, this particular fungus has these genes and it's found in all these groups, but none of these. Okay. Over here, it's got a few different changes in it. So that's how they were able to say that, like, this was co-opted and modified, and the few changes that happen in that gene are the modifications that they're talking about. It's kind of a lot going on bioinformatics wise that this paper did. And so
Hey, Dr. Zahn, would
yeah.
you, oh, before you go out of the phylogeny, would you, or the phylogenetic trees, would you mind going back in and just re-explaining those a little bit? Whenever those get pulled up, it's kind of like my nemesis. I'm always like worried that I'm not quite understanding them enough. So could you just re-explain exactly what we're looking at here?
Okay. So we've got our 72 species that were looked at in this particular study. Okay. This is showing the hypothesized evolutionary relationship between all of them based on genomic phylogenetic analysis. So not just a single gene or anything. They went in and took as many genes as possible, in a lot of these cases, full genomes, aligned those genomes, and then constructed a tree that is the best explanation of how we got these changes in the genomes. Okay, so that's what we're seeing here. This is a phylogeny. Here we're saying there's fungi at this node. And so we've got Rosella, Allomyces, okay? We've got Blastocladiomyces here, the chytrids here, the Zoopagomycota and Mucormycota, so those are our zygomycetes. Then here's our Ascomycetes. You can see that those are monophyletic. Okay. Here's our Basidiomycetes, also monophyletic. Basidiomycetes and Ascomycetes are sister to each other. This was their common ancestor, and it was sister to the Mucormycota, which we think of as zygomycetes. Okay. But we also think of Zoopagomycota as zygomycetes, if you remember. And this is why we can say zygomycetes are actually not monophyletic. It's a fake group. Okay. Because we group them all together when really the closest relative of Mucor is Ascomycetes and Basidiomycetes, not Zoopagomycetes. They share a more recent common ancestor. Okay, so now in this phylogeny, they've also gone in and added some like color coding. Okay, hyphal is shown in green, non-hyphal in blue. Does that make sense?
Yeah. So then my one question about that would be, is how come if we look here, it's hard to see on my screen, it's so small, but where it's Chytridiomycota, how come some of those species have more of the dark, which was the non, right?
Yeah.
How come they have more of that than their ancestors? Is that just because of natural selection that they've selected against it?
Yeah, successive loss
Okay.
of hyphal genes. Yep.
Okay.
And these are predicted hyphal genes. We're not 100% sure on them. Okay. The numbers that you see next to phylogenies like this are called bootstrap values, and they're, in the simplest way, they're kind of like an estimate of confidence in that given node. Okay, so here we're saying, like, between these two different versions of making the tree, we're 99% sure that this node is real and that that's a monophyletic group. Right. So here they're saying origins of fungi, they already had some hyphal genes or predicted hyphal genes in there, but we've got groups that aren't hyphal mixed in. If we look up at our ascomycetes, here's Saccharomyces, okay, and it's a nice little mixture of hyphal and non-hyphal growth forms. So it's a complicated figure. It really is, kind of showing a lot. Copy number. Okay, and so this, in this part here, they're showing the number of gene copies for a given type of gene family. So for cell wall biogenesis and remodeling associated with hyphal growth, here's the gene copy number. Significantly different, p-value is less than 0.05 than other groups. Okay, so it was enriched in these 3 nodes. So what they're saying is that these are the types of proteins: signaling, cell cycle, actin, cytoskeleton, vesicle transport, polarity maintenance, that were enriched in fungi that were becoming multicellular.
Okay, it's a lot to process, but I definitely get it a lot better than I did before.
It is a lot to process. I don't expect you to look at this chart and immediately know what's going on. It takes some time, like reading through the caption over and over and trying to map it onto what you're seeing here. It also takes practice reading phylogenies, which is important in biology. It's why probably every class you've ever had is showing you a phylogeny of some sort. I assume. Can't think of any biology classes that wouldn't bring up phylogenies at some point.
I do have a quick kind of fun fact. So I read this paper and then I'm taking evolution, and the book we're using actually cited this paper as a reference. They were talking about fungi and how they develop in a separate way, and it was really fun that it matched the timing. I don't know. It was really fun.
The book you're reading cited this?
Yeah, it's
oh wow, because it just
exactly
came out in like April 2019, so that's good.
Yeah,
September.
Okay. I was very impressed too.
Yeah, that is impressive. But immediately, this paper was like an overnight classic in the mycology world.
Evolution, I would see what that would be. There's an evolution. It's an evolution class, and the book talks about evolution, and yeah, so I can see why that would be a hot topic there too.
Yeah, the mycology world. I remember when this paper came out, and everyone's like, "Whoa! Did you see the new Nature paper on fungal multicellularity?" It's like, no, I don't have time for that. And then I finally read it. It's like, whoa, that was a ton of work, and it had to have been a ton of work. The analyses they do in here are really in depth, really high quality. The amount of evidence they accumulate to put into one paper is super impressive. They could have gotten 6 or 7 papers out of this, right? If they piecemealed it out, but they stuck it all together for a nice, really good, like big picture overall view of how fungi did multicellularity so different than any other group. Animals are duplicating adherins and cadherins, cell signaling proteins. So, they're getting cells to stick together and talk to each other, and how to organize those cells into distinct shapes for body plans. And it's a lot of duplication, and fungi are just like, nah, that's fine. We're just going to take old stuff that we were doing already and turn it upside down. And instead of growing in, we're going to grow out and just accidentally become hyphal through a series of small gradual changes. So pretty neat. All right. Well, we've got, what, 45 minutes-ish? So I would like to take a look at medical mycology, if you all are ready to move along a bit. It won't take us long. Sound good?
Yeah, let's do it.
All right. So short slideshow, but I am going to give you an assignment related to medical mycology. So let me go ahead and share my screen. Okay, is that working?
Yep. That is definitely working.
Oh, great. So medical mycology, as you might expect, is a pretty important field, but it's one that, like, oddly enough, hasn't gotten tons of attention because it's kind of difficult, because a lot of the mycoses, that's the term for fungal infections in humans, they disproportionately affect people who are immunocompromised or who live in regions of the world like Papua New Guinea. And I hate to say it, but we just have a history in medicine of not really caring that much about those groups of people, not to the same extent as some other diseases. However, this is changing, and people are starting to care about the sorts of people who are getting sick from fungal infections. I know that's a bleak way of putting it, but it's probably not far from the truth. Okay. So we'll just take a brief look at the sorts of infections that happen and how and why they're increasing. I threw this paper, the actual paper, up on our website for you. But this is like a pop science article about it. Fungal diseases are on the rise. Is environmental change to blame? Well, yes. Okay. So that's a paper by Arturo Casadevall and a couple other people. I forget their names currently, but they did show that, and so we'll take a look at that in a second. But depending on who you talk to, what doctor you talk to, you may get myths like this, that fungal infections are rare, or they're really just for immunocompromised people, or just in the tropics, and that fungi can't handle warm body temperatures. That last one is somewhat accurate, and it seems to be one of our greatest defenses against fungi. But in reality, fungal infections are really widespread. They're not all super serious and life-threatening, but they're way more common than you might think. The most dangerous ones do tend to disproportionately affect people who are immunocompromised. So HIV patients, people undergoing organ transplants, people with autoimmune disorders across the spectrum. That's who's really at risk from very dangerous fungal infections. But they're super common, right? So we got toenail fungus, we got athlete's foot, ringworm, candidiasis, right? So all kinds of stuff affecting people all over. So far, maybe 150 species of fungi are known to cause infections in humans, including immunocompromised humans. And maybe 20 or 30 are major threats. Okay. So these numbers are another reason why mycoses are given less attention than other illnesses. Okay. We do have to expend our resources somewhere, and maybe mycoses just aren't a sexy enough topic, right? There's only 20 or 30 species that are super dangerous to humans, okay, but they're all over the place. Okay. So it doesn't mean low case counts just because there aren't very many different species that can do this. Okay, roughly 1.5 million deaths annually worldwide, but a lot more of just really uncomfortable, bad infections that aren't necessarily life-threatening. Okay. So these are on the rise. We've been noticing this over the past 15 years that fungal diseases are becoming more common, and they're showing up in areas where we didn't used to see them. Okay, so this is a map of Vancouver, and this is an outbreak over a 10-year span of Cryptococcus gattii. Okay. Cryptococcus gattii used to just be in Papua New Guinea. Okay. Why is it turning up in Canada?
Migration, probably.
Okay, so it's got to get there.
Yeah.
It's probably not going to make it through the air. So it's turning up in Canada because
it has a carrier vector. Yeah.
People are traveling from Canada to Papua New Guinea. Okay, so the walls that used to separate different compartments of our world into little biomes, as far as pathogenic diseases go, are breaking down because of global air travel. Okay. See the same thing in like plant diseases, right? But these fungi are also adapting to warmer temperatures in these places. So before, it was less likely that this outbreak could happen in Canada because the fungi wouldn't survive there to reinfect humans. But the fungi are evolving to an increase of 1.5 degrees in the global annual temperature. And so as the fungi evolve to that, they're actually evolving to be a little more tolerant of human body temperatures. In the past, and even still now, honestly, one of the major things that keeps us safe from fungi is the fact that we're warm-blooded. Most fungi cannot survive at 37°C. Okay, so this is awesome. Arturo Casadevall here, I went to a mycology conference and he was kind of gave the plenary lecture, and he spent an hour describing this insane, like, crazy Jurassic Park story about how fungal diseases are why there's mammals rather than dinosaurs ruling the earth right now. He's not saying the asteroid had nothing to do with it, but he had some really interesting hypotheses, kind of far out there, but interesting, about how fungal diseases, they do affect snakes, reptiles, way more than they do mammals, and it's because of that internal body temperature. Okay, so conditions are changing around the world, and fungi are actually evolving resistance to higher temperatures. So you can expect that fungal diseases are going to be more of a problem for humans going forward. We've actually already seen that. Some other factors that might explain the dramatic increase we've been seeing in mycoses over the past 15 years are that new fungal diseases are actually emerging from the permafrost that's been frozen for 30,000 years. So as the permafrost in Siberia thaws, we're getting like new strains of anthrax out of there, but we're also getting new fungal diseases that humans haven't been in contact with for tens of thousands of years. We are getting a lot of population growth in regions with endemic pathogens, so in these tropical zones. That doesn't help, and increased human density always means increased disease load. Right? And HIV is still a growing problem, particularly in sub-Saharan Africa, right? 1 in 4 people have HIV in some regions. That's insane, and so fungal pathogens are going to be a major problem. All right. Now,
John,
yes.
sorry, speaking of immunocompromised, what about like cancer patients? I mean, they're not your typical, what you would think of, but I mean, when you undergo cancer treatment, you're severely
Oh, yeah.
compromised.
Yeah,
I
so
mean, it's interesting to me that it's not an interest, you know what I mean? Because I'm thinking Western
I'm not
world,
saying it's not an interest.
well, not as much as it should be, yeah.
It's not prioritized. It's really not. Because I think it's maybe like a legacy effect. People who are teaching at medical schools,
Mm,
when did they get their degree?
many years ago, right?
Yeah, so
Yeah.
they
Yes.
got it a long time ago. A lot of them, frankly, before like modern molecular methods for kind of addressing these issues. And so they're taught what they've learned, and
Right.
so
And this is not a focus. Got it. Yeah.
yeah, it's going to change. It has to.
It has to.
Yeah, and so it is. A friend of mine was a computer programmer. He was 55 and decided on a whim he wanted to be a doctor, and so he went back to school and like took all his classes.
Hey, I know.
Got
That
into med
feels.
school. This guy is incredible.
That's amazing. Yeah.
He actually got into med school and he got there, you know, at like 57 years old. Shows up, good med school, UMKC, and he knew that I was into like microbiome kind of stuff. I was still an undergrad, but I was really getting into like next gen sequencing and microbiome. So I was talking with him about that, and he said, "Oh yeah, we talked about that in med school. We had 1 hour lecture on human microbiomes, and that was it." Like, are you kidding me? It's like, nope, just an hour. It's like, there were like 10,000 papers published this year on this stuff. Like, yeah, it's just kind of a legacy effect of the teachers. It's not like they discourage you from learning that stuff.
Learning,
Oh,
right?
yeah, but it's currently, at least in the recent past, has not been a big focus of medical programs. But I don't want to be a downer. It is changing, and we're getting pop science articles about, killer fungi coming out of the tropics, and so this will be a focus in the future. Okay.
Do you know even is there even testing done? I mean, how do they? When you go to the doctor, did they just like look and they're like, well, it's not a bacteria, so we're just gonna test for fungi, or is that even a thing happening? I don't even know.
I've just from personal experience going to the doctor, and even to the vet with my dog.
Right, I know vets do a lot.
The vet was actually more interested in testing whether it was a fungal infection in my dog's ear than the doctor was for me. And the vet was just like, well, I'm gonna smear some of this on a slide. And he looked at it and he's like, well, it's bigger than bacteria. It's some kind of yeast. So it's probably a fungus. We're going to give antifungal stuff. And so it's not like out of their mind, but it's definitely not the first thing that a family practitioner is going to think of is fungal infection, unless it's one of the big ones, right? Like ringworm, you know that one. Athlete's foot, you know that one. Candida, here's another one. So this is common in like infants, people who eat too much sugar, honestly, that is a trigger for candida. It wants to live as a yeast, okay? And it's always living in your mouth. You've got it in your mouth right now, I promise. If I swabbed you, I'd be able to culture candida from you. But it's fine there as a yeast. It's actually doing a good thing. It's kind of taking up space, occupying a niche that harmful bacteria or fungi could live in. But it's dimorphic, so it can turn hyphal and grow filamentous. And when it does that, that's a problem. That means it's growing into your flesh, right? Eating you. So it's a facultative pathogen. It's ready to take advantage of you the first chance it gets, right? Cryptococcus. This is, again, until the past 20 years, this was limited to Papua New Guinea, tropical zones in that region. but now it's taking over the western United States. Okay, so Cryptococcus, it's a bad human pathogen, gets in your lung. It actually has complex life cycle associated with your lung and your bloodstream. Aspergillus, we've seen this. It's probably growing on your bread right now. It's an ascomycete. It's just opportunistic. You're breathing in spores all the time when you walk around like a moldy old house or have a moldy sandwich on your counter, but it can become pathogenic. And then mucor, the medical doctors haven't heard yet that zygomycetes are polyphyletic, so they just call everything mucormycosis. But it's basically any disease caused by a zygomycete gets the name mucormycosis. They're relatively rare, but can be very serious. All right, so just a quick look at these 4 main problem fungi. This is Cryptococcus gattii. Okay, it's a jelly fungus in nature when it's out there hanging out in the tropical woods, or apparently in Washington now. That's not going to affect you at all in its teleomorph form. It's the anamorph form, the asexual form, that can infect your lungs and brain. Okay, so yippee. Trigger warning on this next slide. This is mucormycoses. All right. So all these genera here, Rhizopus mucor, Rhizomucor, Cunninghamella, Apophysomyces, okay? They all get called mucormycosis in the medical community. And if they are able to take hold of you, they're incredibly, incredibly pathogenic. So look at this. I mean, it's just eating away your nose, your eye, your entire scalp. Some of them can actually digest parts of your bone. Here's a man who, you can click on that if you want, actually lost his entire face. So he had a face transplant, but this was because of a zygomycete infection. Ate away chunks of his bone, his eyes, his nasal cavity. Okay. So nasty stuff. Aspergillosis.
Dr. Zahn?
Yeah.
Sorry, I just have a curiosity question on that. So I feel like there are a lot of people who are immunocompromised, and you said we're always breathing in mucor, right?
Yeah.
So I mean, like, why don't we see this more often? I don't know if you know the answer to that, but it just seems like it'd be more than rare if it's all around us and people are immunocompromised. You know what I mean?
I don't think it's being, for this, just immunocompromised is enough. I don't think we know why one person gets it and another doesn't. Some current hypotheses are saying that it is genetic, if that makes any sense. So something about your genes, the way you're expressing this, makes your body really have a hard time fighting off these particular types of fungi. So even immunocompromised people may have systemic resistance to this just because of the fact that you're human, you've got a high body temperature, and that kind of stuff. Cell receptors, that kind of thing. I don't have a good answer as to why some people get it and some don't, but talk about unlucky.
Yeah, I feel like genetics is usually the answer when no one knows what the heck's going on.
Exactly, and it's probably the right answer, to be honest, but
I wonder if it has something to do with the bacteria on our skin also. Maybe it must have some genetic aspect to it, but I wonder if that has something to do. And they just don't successfully fight off the fungi. I don't know.
Quite possibly. And in your reading assignment this week, it covers the link between fungi and bacteria in the human microbiome, with the caveat that the paper basically just says we have no idea
we don't
what the link is, or if there is one, we need to do tons of research on it. And that's still the case. That paper was from 2013, and I would still say that we have no idea what the link is between major components of our microbiome. We're still in the infancy of that research. We're getting there. I mean, we've got cool success stories for microbiome research, but nowhere close to having a comprehensive theory of how microbiomes work to modify human diseases.
How fast does that fungus grow when it comes to that?
Very. I mean, think of how fast it takes over your sandwich.
Okay.
So a matter of days.
Yeah. That's impressive for that microorganism to move that quick.
Yeah, it's terrifying for us.
Yeah.
But good for it. Congratulations, I guess. And it's really difficult to treat. Same thing for like aspergillosis. This is causing like sinus and lung infections. Immunocompromised people, again, are at the greatest risk, but some people are essentially born unable to fight off aspergillosis and have like chronic lung infections from this, even though their immune system is otherwise really fine. Part of the problem may be that fungi and animals, humans, are so biochemically similar to each other, right? And then candidiasis, we briefly mentioned this one already. It is a major component of all these aspects of your microbiome. It's like all over the place. Lost my mouse cursor. Okay, so it's in your lungs and skin, oral, bowel. It's living all through and on your body. And people at greatest risk for this are actually hospital workers because they're in close contact with sick people all the time. Okay, so we got problems treating fungal infections. It's hard to make antifungal drugs. How come?
Are they just evolving too quickly?
They do evolve really quickly, especially in a hospital setting.
This might not be the answer you want, but I think it could also relate to the fact that, like we talked about, how like they're a lot more rare than a lot of the other conditions that people would get. So I think funding might be a lot harder to get when it comes like these. I don't know. It's just an idea.
No, I think you're right. The same thing's happening for antibacterial drugs too. There's no profit motive.
That's where I got my idea from.
Yeah, there's no profit motive for private companies right now at all to make new antibiotics.
Is it also kind of the idea with the septae we were talking about before, where you get a drug in a fungus, it realizes, oh my gosh, I'm dying. It can literally just shut off that, kill that end of itself and still survive and move on.
Yeah, that might. So you can't just apply them topically. They do tend to have to be a more systematic. So you'd be applying this topically also, but taking antifungals as well. I think a major problem is that our pharmacy industry relies on solely profit motive to generate new things. We don't subsidize it based on where the current need is for developing diseases or anything. There's some interesting ideas for maybe trying to solve that problem, like to offer government grants for like developing new antibiotics. So private companies would compete to cure an emerging disease, and then they get $3 billion of taxpayer money if they get it, right? So that would kind of take the risk out for them. Another proposal is that we take the profit motive out of it entirely and make it a public mandate to where our taxes are directly funding the prevention and treatment of emerging diseases. I don't know how much political willpower there is for that in modern political climate, but right now something's got to change because we're not creating new antibiotics at all. One of the reasons, of course, is that bacteria are just evolving resistance to them so fast that it takes, $1.5 billion to make a new one, test it, and make sure it's safe and effective. And then in a year or two, it doesn't even work anymore. right? So no pharmaceutical company in their right mind is going to go after new antibiotic drugs, right? You get maybe a year and a half of profits. That's not enough to recoup your investment. Okay. Another problem that's especially hard for fungi is that they're actually harder to create drugs for than bacteria in the first place because they're eukaryotes and because they're sister to animals. What's that have to do with anything?
Well, in bacteria, you can target the peptidoglycan and direct medication straight to bacteria and not kill eukaryotic cells. But how do you do that when all of them are eukaryotic?
I mean, even bacterial like DNA replication is different than eukaryotic. So, I mean, there's tons of targets you can go after for bacteria. In fungi, you've got
the cell wall, I guess.
Cell wall. You've got the fact that they don't use cholesterols. They use I'm brain farting. Well, that's embarrassing. Ergosterol, that's it. So instead of cholesterols, they use ergosterol, so that's another target. Well, that's about it. What else do you target? Everything else that you would target would be something that's essentially identical in the human cell. So it's really tough to find antifungals that actually work. And then we've got fungi now that are becoming resistant to heat and are multi-drug resistant. This is a problem. Okay. But not all fungi are bad for humans. We got what we call the mycobiome, cheekily named there. But fungi are commensals. They live in and on our bodies, and they're doing important stuff for us. Candida especially is actually, although it can turn pathogenic, it's actually probably a pretty important part of our microbiome, okay? We've got stuff living obligately in our lungs. We used to think our lungs were sterile, which doesn't make much sense, but we do have resident communities of bacteria and fungi that live in there, taking up space, might be helping us out, preventing infection, but sometimes can also turn bad. So I want to talk just real quick about some of my research students I had recently go through. They have both graduated and are in dental school now, but when they were here at UVU, they decided to do a fungal microbiome project. And so they were looking at oral fungi, and they had people basically swish sterile saline, then they collected it, we extracted DNA, amplified some fungal marker genes, and that was the plan at least. Then they switched to bacteria halfway through their project. Why? Any guesses? Has anyone read the paper for this week yet? Fungi are harder to study than bacteria because the databases we have aren't as good. Okay, so we did amplify fungal DNA from these samples along with bacteria, and we just couldn't get any good results from the fungi. Maybe it was lab technique, maybe it was the databases, but it just wasn't quite working. At any rate, the microbiome is definitely an emerging area of human health. It's not as numerous in terms of number of species or abundance of species as bacteria in the human microbiome, but we're starting to think that they're pretty important components. Okay. So when you read through the paper, you'll kind of see a glimpse of human microbiome research. Okay. And what I want you to look for in the paper is the interaction with the immune system, fungi as treatments for diseases. That's actually an emerging paradigm right now. What makes a fungus go from commensal to pathogen? That we still don't really know that. how they interact with the bacterial microbiome, and if you get them out of whack, dysbiosis, does it make you sick? We noticed that in bacterial microbiomes for sure. Things like Crohn's disease are inextricably linked to dysbiosis of gut bacteria. We also notice fungal patterns with Crohn's disease that's brought up in the paper for this week, but we don't know what's going on there. Okay. And of course, a couple more classes of medical mycology is just spores being an allergen, kind of no different than pollen. But roughly 6% of the population is allergic to fungal spores, which sucks for them. And they may not even know it until they get a doctor that thinks to test for, Alternaria and Cladosporium as allergen agents. So it may take a little while. You run around with itchy, watery eyes for 3 years before you finally see an allergen specialist who finds out, oh yeah, you're allergic to the air because fungal spores are everywhere. Okay. And then last but not least, mycotoxins. So these are pretty bad. Claviceps, I don't know if you're familiar with this, it's ergot. It grows on rye and cereal grains. Those are the claviceps, essentially like teliospores, basically. It's making basidia on the structure. If you eat that, this happens to your extremities. Well, that's real great, especially before people knew to look out for claviceps on rye and when rye was an important cereal grain. So it makes your hands and feet fall off. It basically shuts down blood circulation. Okay. It also makes you hallucinate and see demons and stuff. So that's really exciting. Then we got Amanita. There's our destroying angel mushroom. It grows here in Utah fairly abundantly in the mountains. That will definitely kill you, so just stay away from it. Okay. Now, your assignment is to read the Human Microbiome review paper. It's a bit older, so if you have questions, I can point you to newer papers, but this is still probably the best overview of human microbiome research. And then you've got a new assignment up on Canvas. It's to go to the CDC website. I've got a link there on Canvas, and I want you to make a PowerPoint slide about a fungal infection. Okay? So details for that are up on Canvas. Let's see if I can bring up the assignment. Create a PowerPoint slide about a human fungal pathogen. All right, so there's your link. Go to the CDC site here. It's got types of fungal diseases. So pick one of those that's interesting to you and make a slide. All right, so here's candidiasis. You can see vaginal candidiasis, invasive candidiasis, antifungal resistance. Just condense all this information. Use some outside resources for pictures if you want. And just make basically like a little informative slide, one page, not a whole slideshow, about that disease. Okay. As you're reading the paper from this week, keep in mind it was from 2013 and it's an overview of the emerging science of human microbiome. Here I've gone to Google Scholar and I've just searched for fungal pathogen plus human, and I've set it to be just for the year 2006. So this is the year I started my undergrad, well, after failing out a couple times. This is the year I really started my undergrad as a biology student. There were 1,500 results from that year about human fungal pathogens. So 1,500 papers were published that have those as keywords. You can kind of glimpse through those. If I click on just the year 2020 so far, 4,620, and it's only September. Okay, so you can kind of see through time how this is becoming a more in vogue area of research as people start to learn more and as fungi become drug resistant and start being found in new places. This is definitely getting more attention. And it's not like these papers were just written overnight, right? Each one of them probably represents several years of hard work from people. So there is a lag in this also. There's a lot of ongoing research right now, and I expect the year 2021 will have another 5 or 6 or even 7,000 papers published on fungal diseases. So even though we are giving a kind of short shrift in this class, really just one lecture and an assignment, it's something to consider if you're going into the medical field. Insist on learning more about it once you get into med school, and just keep in mind the life cycles of those fungi and look up the papers. All right, any questions for today? Nothing? We got 5 minutes left. Have I dropped off or something? I've been talking to nobody this whole time.
No, we're here.
Oh, okay. Okay.
No, I got
They were
rid of
just
all
off
my ink stamp questions at the beginning, so I don't have any more left.
Okay. All right. Okay, well, have a good one. Enjoy the review paper. It's pretty easy to read compared to the last one. And I will see you next class.
Thank you, Dr. Zahn. We'll see you guys.
Thank you.
Thanks,
All right,
Dr.
later.